Good morning. Can you hear me?
Yes, madam. Good morning, madam.
Right. Good morning. Where's your batch? Is he there? I see about 50 of you are here, no? So what about the situation of revision lectures? How many roughly participating?
Yes,
You must
madam.
have had two, no? Previous ones?
Yes, madam. We had about 100 participants, madam. 110 we had.
About 100, no?
Yes, madam.
Right. Let's see. Right. Shall we start? Can you see the screen?
Yes, madam.
No? Right. So this is today we will be talking about something related to embryology, right? So to revise embryology, you should have gone through the past papers and identified areas where questions have been asked, right? So when you take the essay paper, right? For every question, we have 5 questions now. So for every question, there's an embryology component. And for the MCQ paper, MCQ and SPR paper, paper 1, for MCQs, you get about, let's say again, about 6 questions based on embryology. About 3 of them will be on general embryology, where we will be talking about either gametogenesis, or maybe about the first week, second week, fertilization, all those things, right? Cleavage, granulation. And the remaining 3 are basically based on your systemic embryology. So we might ask you about the cardiovascular system, respiratory system, gastrointestinal system, so on and so forth, right? So nobody can train you to answer MCQs. What you have to do is you have to train yourselves by doing MCQs, right? So therefore today I will be talking about basically on what type of questions that you will get as a part of your essay paper, right? And if you collect all 5 questions, the total amount of marks that will be awarded for embryology will be approximately 100. If you have carefully gone through the past papers, you may have seen that, right? So under each question, we will be asking you about something, right? So let's take it one by one. We might ask you something about general embryology that all of you know, no? Right? So what are the things, what are the questions we might ask you on general embryology? We generally ask something about the general embryology in the section of upper limb, right? Or in the section of upper limb, we might sometimes we might ask you about the development of a particular bone, right? Maybe about the humerus, most of the instances, or something again related to general embryology, right? And in the part of the lower limb, if there's a lower limb question as well, there also we can ask about the development of a bone. Let's say the femur, where you have to talk about ossification centers, type of ossification, this and that, right? So other than that, the rest of the general embryology questions are most of the time asked either in the upper limb or lower limb, right? So what are the things that somebody might ask you, right? So when you analyze each and every question, you will see what type of embryology questions that could be asked in that particular section. So that is one of the things that you have to do during this time as an activity. Take each essay question one by one. We may have asked about something. What are the other embryology questions that you can ask from that particular section? Because it has to be related to the main section that we are going to ask, right? So if you do that in as an activity, analyzing each and every question, you can almost always guess what are the type of questions that would be asked in each essay question, right? So this type of activity is very important because you get used to the essay paper. Otherwise, most of you will go and and read the essay paper first time at the exam. Then you are not familiar to it. And the other thing is most people think that just going through the questions would be enough, which is a wrong idea. You have to answer questions to the time limit. Let's say per one essay question, you can take about half an hour and answer, right? Comprehensive answer without looking at a book and evaluate yourself, right? So that is a little bit of general information that you need to know, right? So we'll move from one section to the other and see what type of questions that will be asked. Right. So somebody might ask you about gametogenesis. This is, of course, not only in the first part. If somebody asks you, if there's a question on pelvis and perineum, let's say somebody is asking about the uterus and the tubes and the ovary. So there somebody might ask you about gametogenesis, right? So what should you know about the gametogenesis? You should basically know something about the meiosis and the differences of meiosis with mitosis, right? These things you have been learning from your school days now. Basic differences between the two. And the products of meiosis, you know meiosis is in two parts, meiosis 1 and meiosis 2. And when the special features of meiosis, pairing of homologous chromosomes, crossovers, synapses, exchange of genetic material, right? Most important things of meiosis. So such things you have to highlight when you're answering. Somebody might ask what the differences of mitosis and meiosis in general embryology, right? Under the section of a question where they are talking about female reproductive system or male reproductive system, right? So any of these things are possible. So when you take a question, think about all these possibilities, right? And again, somebody might ask you the differences between oogenesis and spermatogenesis, right? Don't forget these things. These are basics that you should know, right? And I think this picture will summarize the basic differences, right? How a primary oocyte will give rise to 4 cells, and only one of them is the mature oocyte and the 3 others are the polar bodies. And in spermatogenesis, the primary spermatocytes will give rise to each primary spermatocyte will give rise to 4 spermatids, either carrying 22 plus X combination or 22 plus Y, right? And when we are talking about the females, female meiosis, do not forget about the extended meiosis they have, right? It's a special feature in female gametogenesis, where you have a long meiosis, whereas such thing is not there in a spermatids. Once you begin the formation of spermatozoa, it will go on like this without a stopping, without periods of extended meiosis, right? So those are few basic differences, and morphologically, your primary oocyte will not undergo much change when it becomes a mature oocyte. But whereas your primary spermatocyte, it does not end up here, no. It will get a morphological transformation into spermatozoa, where it develops a head, midpiece, and tail, which is important for the motility, right? You call that process spermiogenesis, right? So likewise, you should be able to compare and contrast spermatogenesis and oogenesis, right? Right. How many of you are here now? 82. Can you please enter your registration number with your name to the chat so that I can mark the register later? Okay? Hello?
Yes,
Can
ma'am.
somebody respond? Yeah. Otherwise, I don't know whether you are listening, you can
Yes,
hear,
madam.
right? And what happened to the other people? I hear only your voice, right? So when I ask something or when I want a comment, please give me that comment. Otherwise, it appears as I am doing it to only to myself, right? The feedback is very important to the function of biological systems. Okay? Right.
Okay, madam.
Right. So these are few things that somebody could ask you. Right? So don't worry about these molecular biology parts and things. Some of these things you may have learned in genetics. So we don't generally include questions based on genetics for your second paper. You might get about 2 or 3 for the first paper. Right? Right. So if somebody asks you about the first week of development, what are the things you can ask you? We can ask you, right? We can ask you the process of developing primary oocytes and the formation of the follicle, conversion of primary follicle into a secondary follicle, and after that you get Graafian follicle, so on and so forth. The changes you observed. Observe at each level, right? So you might ask you something like that. You have to be ready to answer any question that would be asked. And sometimes we might ask you the developmental changes that occur in the oocyte during the ovarian cycle. Again, the same thing, right? So that is some of the things that we can ask you. And we can ask you about fertilization. right? Requirements of fertilization, right? And things that happen as a result of fertilization, right? The main results of fertilization. I'm highlighting important parts in this, in your textbook, right? So, what are the important things that happens as a result of fertilization? Restoration of diploid number of chromosomes is number one, right? Your oocyte and spermatocyte, both of them are haploid cells that has 23 chromosomes and in amount of DNA, right? When they combine, you get 46 chromosomes or 23 pairs and 2n amount of DNA, right? So diploid number of chromosomes will be established and it determines the sex of the individual, whether it would be an XX or XY, right? And thirdly, it initiates cleavage. What is cleavage? Can anybody tell me?
Division of cells.
Yeah, it initiates the division of cells, which is known as cleavage. And if you have gone through past papers, you might have seen, sometimes we have asked, describe the process of cleavage or what is meant by cleavage, right? So you have to tell us how it is initiated. It is initiated definitely by the fertilization. And if fertilization does not happen, oocyte generally degenerate within 24 hours of ovulation, right? And you have to talk about how the cells divides. One cell divides into 2, 3, 4, like the morula formation and the blastocyst formation, the stage at which it is implanted in the uterine cavity. So this is one of the sections that we might ask you, right? So let's see. Yeah, this shows the process of cleavage, right? And somebody might ask you blastocyst formation, right? This morula, right? This is your morula, no? Right? This one will, the cells in the morula will separate into two groups, inner cell mass and outer cell mass, right? Forming the blastocyst. Inner cell mass will form your embryo proper, whereas outer cell mass will form your placenta, right? So even though this textbook contains difficult terms, new terms, not unfamiliar terms, you can use your own term to describe them. The only thing is you have to remember a few scientific terms, that's all, right? So no big deal. So somebody can ask you the process of implantation, right? What happens to the trophoblast, how it divides into cytotrophoblast and syncytotrophoblast, so on and so forth, something like that, right? I'm moving a little fast because what I'm doing is to highlight you all what are the different questions that we can ask you and how are you going to answer them, right? One other thing I want to tell you is okay, sorry for the disturbance. You can hear me now, no?
Yes, madam.
So, because for embryology revision, doing revision on only one section is not fair, I think, because you can get questions from anywhere. No? So I am just trying to give you an impact on how to answer these questions. And most of the time, you people answer histology questions with diagrams. But that you know, because if you don't draw histology diagram for histology, you will lose certain amount of marks. But none of you, only few of you bother to draw diagrams in embryology, right? Because you do not know the fact that when you draw a diagram, you can always use less words to explain in the text. And it is always easier for you to explain when you are comfortable because you are deferring your own diagram and answer. So most of you don't. But my advice is to draw a diagram wherever possible. If you read the instruction on the top of the paper, we generally say illustrate your answer wherever possible, because always illustrations will help you to tell something, because this is anatomy which is easily explained by illustrations. So keep that in mind. If you draw a diagram, you will always be benefited by it, right? So please keep that in mind and try to make your answers easy to read, nice ones with diagrams that are self-explainable. Then always when within the text, you can always ask your examiner to refer the diagram. If your diagram is a complete one with fully labeled and necessary details, no harm doing it. Right? And it will save time also. Quite a lot of time when it's a struggle with time. No, I'll try to answer 5 essay questions. So you can always save your time by doing things like that. Right? Again, in embryology, we can ask you the morphological changes in the endometrium during different phases of ovarian cycle. Right, so we'll move on to the other one. Second week of development. The bilaminar germ disc, right? So you have to mention what bilaminar germ disc is. What are the 2 layers of cells, epiblast and hypoblast? And where is your amniotic cavity? And where is your blastocyst cavity, right? And the changes in the trophoblast, formation of cytotrophoblast and syncytium, right? So when somebody asks you about the second week, that's all you have to write. And towards the end of second week, you can mention a little bit about this changes in the syncytium, where the spaces appear or maternal lacunae will appear, and as the syncytium grow further, it will invade the blood vessels and blood will come up and fill these spaces, establishing the uteroplacental circulation. This actually happens towards the end of the second week, maybe in around 13 to 14 days like, right? So remember about the cavities, amniotic cavity, and how your blastocyst cavity becomes the exocoelomic cavity, right? This is just change of names with a little change in morphology, right? So you can see what I said. This is, I think, about 12 or 13 days. How this is the time where the uteroplacental circulation start to establish. So that is one of the important features in the second week of development, right? And towards the end of the second week, again, you can see the amniotic cavity here and the exocoelomic cavity or the yolk sac, primitive yolk sac you had. And you can see the formation of extra embryonic coelom outside to this and how this happens. Read a little bit of theory and you will be able to revise it. Okay? Right. So those are the few things that you will have to. So you can see this is maybe 13, 13 day. Amniotic cavity is there. Your yolk sacs become the secondary yolk sac now, and you can see this formation of extraembryonic coelom, which is also called as chorionic cavity, right? So there are only few things, but you must always not to forget to highlight the important things, right? Right, so that is about our second week. And when we are talking about the third week, what are the things that we have to talk about? In third week, you know that the two layers will be replaced by a third layer, right? So you should know the phenomenon, what actually happens. This is the period where your ectoderm, endoderm, and mesoderm is established. So formation of primitive streak and migration of cells from either side in the epiblast into the tissue underlying. First, it will go and replace the cells in the hypoblast, right, forming the endoderm. And then it will be, some cells will be deposited between the ectoderm and endoderm, forming the mesoderm. And the cells remaining on top will become the ectoderm. So as you go on during the gastrulation process, what happens is the epiblast replaced all the other existing cell layers and they differentiate into ectoderm, mesoderm, and endoderm. So in your answer, you have to highlight that, right? So gastrulation is the thing that the most important one that you have to describe if you were asked about the week, right? Right. So third to 8th week, I'm not going to talk about details because you know it is the period where your organ systems starts to develop, right? And as a result of that, you establish your respiratory, gastrointestinal, genitourinary, cardiovascular, everything, right? So because this is the period of system formation and establishment, congenital anomalies are common during this period, right? So in case sometimes somebody might ask you, right, to write short notes about 3rd to 8th weeks. Similarly, 3rd month to birth, where the fetus enlarges in size, right? So in the first part, division will take place and systems will establish. And from the 3rd month, the basic systems are now established. What you have to get is the growth and maturity during this period. So those are the things that you have to highlight your answer to give an idea to your examiner that you have understand the concept of it, right? The difference between two periods is that. So that is why 3rd to 8th weeks, you will get congenital anomalies. Whereas from 3rd month to birth, congenital anomalies are rare, almost non-occurring like, right? The reasons behind it. Right. So, from there onwards, I am not talking some of these things because it's very rare that we can ask questions on these sections. From muscular system also, we generally, now we are moving on to the systemic embryology, right? Yes.
Madam,
Yes.
small doubt. Madam, what if before third week, now this during this gastrulation process, what if the cells don't migrate? And what if that process didn't happen? So it will directly lead to abortion, no, madam?
Yes.
So
There's
in
no
case
question
if they
about it.
ask us about the gastrulation, so as a
The embryo
madam,
cannot proceed from thereafter, no? If gastrulation does
hello,
not take place. Can you hear me?
yes, madam,
Yeah, if gastrulation, your problem is if gastrulation is not taking place.
yes, madam,
Right? So there will be no formation of 3 germ layers.
yes, madam,
Your endoderm, ectoderm, mesoderm will not be there.
yes,
Right?
madam,
So if there is no mesoderm, your cardiovascular system will not develop.
yes, madam.
Right? So there's no heart to beat. During early prenatal period, it will be detected. Right? You
Okay,
do regular ultrasound scans now. So it will be, if it is third week, it can spontaneously get aborted.
okay, madam.
So that's what happened.
Okay, madam.
Madam, what about neurulation?
What about relation means what is your specific problem?
No. Is it a essay-based question? Neurulation.
Yeah. Anybody could ask you anything from your syllabus. Right? So what are the things? I think the new relation of we have asked recently. Right? In
Yes.
one of the previous recent papers, it is there. Right? So I am
Okay,
just
madam.
highlighting certain areas that might come. Don't get the misconception that I might miss something when we go on. Right? So just because I didn't discuss about it, don't think that it will not come in your paper. Right? Sometimes I might also miss certain things now. We can ask anything from this textbook other than the molecular biology part. Right? There are molecular biology, there's a molecular biology part in each section now. So we do not go into that extent. So other than that, anything that is taught in your syllabus can be asked as a question. Neurulation, of course, yes. Right? So you have to describe the process, the abnormalities that could result if it does not take place in the normal way, right? And the structures or derivatives that take place, and the approximate age of development that these things takes place. Clear?
Yes, madam. Thank you, madam.
Okay. Right. Right. So muscles, I don't think that we have previously asked about the development of a particular muscle or muscle group. Right? I don't remember that we have asked such a question. But again, anybody could ask that too. Right? So when you are talking about the muscular system, we talk about the development of the mesoderm. You should know what is paraxial mesoderm, what is lateral place mesoderm, and what is intermediate mesoderm. And the structures that will be formed by each of these 3 parts. Right? And if you remember your paraxial mesoderm, you have sclerotome, myotome, and dermatome. Right? There is segmental origin. And what are the structures given out by the sclerotome, dermatome, and myotome? You should be able to explain, because somebody might ask you development of the paraxial mesoderm. Right? The chances are less that somebody might ask you about the development of a single muscle, let's say biceps. So in that case, even if you ask that, this general part should be there. Haritha? I hope you get that message. You can formulate your own questions when you are familiar with this, right?
Excuse me, madam.
Yes.
There was a question, describe the embryological development of paraxial mesoderm.
Yes.
Like that means, madam, we'll have to tell from the gastrulation till the end of the
Yes,
paraxial mesoderm.
definitely. Yes. You have to tell first how the mesoderm develops.
Okay, madam.
In the segment, the differentiation of mesoderm into 3 parts, and then you will focus on the paraxial mesoderm.
Okay, madam. So if we go to write from the two-layer development, it's unnecessary.
You don't have to write about 2-layer development. If somebody asks you about the development of the paraxial mesoderm, you can start from restoration.
Okay.
Right? So that is one of the very important question. Most of you people sometimes do not know the limit that you have to answer and where to begin and where to stop. This usually happens with embryology. Sometimes you write lot of unnecessary details, right? So let's say somebody asks you the development of the ventricular system of the brain, right? You have to talk about the neuroectoderm, neural tube, neural groove, neural tube, so on and so forth. Just a basic description. You need not to have to go to town about the initial thing, right? But it has to be there, right? For example, when you are talking about the development of the spinal cord, you will have to go to town about it, right? Let's say we have to describe about the matrix cells, all these things, even tiny, tiny details, or maybe about the neural cells, just a bit mentioning. But you don't have to write all these things through the general description of development of the ventricular system, right? So this happens, I have seen most of the time with the neuro question. When we focus on one thing and ask about one thing, you don't know the extent what you have to write. Use your common sense. You have to highlight the area that is being asked. And to facilitate that, ventricular system just not come from somewhere else, no. So you have to write a little of the formation of the neural tube. But no, we don't need a lot of details. And bit about how it start and when it start, right? In embryology, in any section, you should know the gestational age of common structures development, right? And you should know from which germ layer it develops. And other than that, just a basic outline of how it happens. You get my point?
Yes, madam.
Right. So accordingly, in case if somebody might ask you about a particular muscle, right, depending on whether it's a limb or axial muscle of the body, slightly differs, no? So you should be able to organize your answer according to the question that is being asked, right? I don't remember that whether we have asked much questions about on this section, but of course we have asked about the development of the paraxial mesoderm somewhere in somewhere, right? Right. So we'll move on to the body cavities. Formation of body cavities is a common question or common section that we can ask you, right? So in that case, like the question that you have asked previously, you will have to mention a bit about the primitive body cavities and how the primitive body cavity is replaced by a definitive body cavity, right? If the question was generally on the development of body cavities. Again, I don't think that we have asked such a question in the past, but somebody might ask you. So you have to start from the most primitive stage where you have the yolk sac, right? And describe about fate of these cavities. What happens to your yolk sac and how your intra-embryonic body cavity develops as a result of this lateral folding. The yolk cell will be gradually absorbed into the gastrointestinal tract. And this is again a place where your diagrams are very useful because this process is very difficult to be written in words, right? Whereas if you draw this A, B, C, D or so on and so forth, this is A, B and this series, anybody who will look at this will know that you know the thing. In essay questions, most of the time what we check is we want to find out whether you know it. Your expressions may be different. The way you express this, way you write it may be a little different from one person to the other. But if you, how far you are able to explain your examiner that you know the stuff is the most important thing, right? So this is one of the instances where the diagrams are very useful to you, right? Right. So we have to describe briefly about the formation of intraembryonic coelom, which will be later subdivided into your peritoneal cavity, pleural and pericardial cavities, right? So initially you know that this is your thoracic cavity and peritoneal cavity, and the partition in between the two mostly done by the septum transversum, and in addition to that you have your esophagus there and two cavities which is open on either side, pericardio-peritoneal canals, which later filled by a membrane, right? So when you are talking about initial partitioning of the thoracic and peritoneal, abdominal and thoracic cavities, you are indirectly talking about development of the diaphragm, no? This is what we are going to tell now. You have the septum transversum that is there from initially, and you have the esophagus and the mesentery of esophagus there, and dorsal mesentery of esophagus and the two cavities, pericardio-peritoneal canals, right? Then which will be closed by pleuro-peritoneal folds. And the four components of the diaphragm. Development of the diaphragm is frequently asked question, right? The septum transversum is here, esophageal mesentery is here, and the pleuroperitoneal membrane is here, and you can see the contribution from the peripheral body wall. And again, somebody might ask you the embryological basis of nerve supply of the diaphragm, right? Because the diaphragm get this nerve supply depending on which structures it develops, right? You talk about the phrenic nerve, and you know where the phrenic nerve comes if you look at this diagram, right? Where your thoracic cavity is divided into the pleural cavity and pericardial cavity by this two folds, pleuropericardial membranes, which grow and unite in the midline, and you have your phrenic nerve located in this fold, right? So phrenic nerve supplies the whole motor supply and sensory supply to the center of the diaphragm, right? Because the septum transversum is just below this, right? Whereas the contribution from the peripheral body wall gives rise to the contribution from the intercostal nerves, which is sensory to the periphery. So when you ask about the development of the diaphragm, again, mostly you will be talking about the diaphragmatic hernia, congenital diaphragmatic hernia, right? foramen of Morgagni and foramen of Borderleg. That are the common ones that would form. So your initial defect, foramen of Morgagni, is an anterior defect, just behind the sternum. The defect is between the sternal slips and the costal ones, origins of the diaphragm, whereas foramen of Borderleg is located posteriorly, right? So diaphragmatic hernia is devastating when you have it on the left side because there's no liver to protect it, right? And at the same time on the right side, it's rare because both the diaphragm, this part of the diaphragm and the liver develops by the contribution of septum transversum, which is there, right? So likewise, important features about these things. The facts that tells your examiner that you know this is important to highlight, right? Okay, we'll move on to the cardiovascular system. And this is one of the systems that where we ask you most questions. But I think recently we haven't asked, no? Somebody might ask you the development of the heart, right? Because it is one of the earliest structures that develop, right? How the heart tube is developed, right? You can see the two dorsal aortae, right? And two tubes on either side with the fusion, with the folding of the, most of these things, the orientation of bodily systems take place in a normal manner if the lateral flexion of the embryo takes place. You can see most of things are led by this lateral flexion. For example, incorporation of the yolk sac into the body, into the gastrointestinal system, and union of two heart tubes together, right, to form a single heart tube, and how the two channels come together, right? All these things are facilitated by the lateral flexion. So what are the things that we are going to talk about the development of the heart? Right? This is a very complex section, right? We will just see what are the things that somebody might ask you. I'm looking for a particular diagram. I don't know whether this book has that diagram. Certain books, when they develop and when they become advanced, certain basic diagrams are not there, right? So I wanted to check in your textbook. There was a diagram, something like this, that shows the external appearance, right, of the heart with atria behind and ventricles in front. And you have the atrioventricular canal here and partition between the aorta and the pulmonary trunk here, right? And the ventricular system developing from here. There was a basic diagram, outside view and inside view. How you divide into different parts, right? Primitive atria, left ventricle, right ventricle, so on and so forth, right? So you should know what are the parts that develops from the primitive atrium, primitive ventricles, truncus arteriosus, cornus cordis. I hope you remember these terms, right? Right. So similarly, structures that give rise from each of these component, right? For example, when you talk about the mature left atrium, let's take one example. How the mature left atrium is formed. There is a part from the primitive left atrium and a part from where? What is the other contribution?
Cardiac cushion.
Left atrium, if you take it as a cavity, this is a left atrium now. So part of the left atrium is the original primitive left atrium. Right? What is the other part? Other part is the part incorporated by absorption of the pulmonary veins into the wall of the left atrium. Right? Remember? Revise these things, right? Similarly for the right atrium. Original rough part and the smooth part. Right? Original part is rough generally. The primitive atrial components. And the secondarily incorporated parts are smooth walled. Right? So somebody might ask you development of the left atrium and right atrium. So this contribution from different parts is one of the important things if somebody might ask you a question like that. Okay? Right. So revise what you know about formation of septa. There are two methods by which the septa are formed: by proliferation of cells like this, or by the expansion of the neighboring cavity. This is sort of a, first one is the active formation, because actual self-proliferation will take place and the two ends fuse, either in this manner or that manner. Or in the other one, a septum is formed by the expansion of the neighboring cavities. So sort of a passive one, right? So if I remember, I haven't done embryology of the cardiovascular system for a long time. I think the proliferation, example of septum formation by proliferation, the best example you have is the septum formation in the primitive atrium, where your septum primum and septum secundum develops. And formation of a septum in the second manner, due to expansion of the neighboring cavity, the best example you have is the interventricular septum. It develops as a result of expansion of the two ventricles, right? Right. We'll talk a little bit about the commonest question that anybody could ask you from this entire system, right? Is the formation of septa in atria, right? So you know about the two septa, septum primum and septum secundum, right? The basic, most important thing about these two septa, none of them are complete septa, right? So you know primary septum primum comes up here and originally there's a gap between the inferior end of the septum primum and the endocardial cushions. This is the area where you develop your tricuspid and mitral valves, right? And this orifice is ostium primum. And later, this will come and fuse with the endocardial cushions. And before the fusion is completed, there will be perforations that are appearing in the upper part, and there is another orifice which is known as ostium secundum will be formed. So that is why we call septum primum never becomes complete, because before it fuses with this and separate these two separate entirely, there's a secondary orifice appear here, right? And septum secundum also, which appears to the right of the septum primum, will go and grow downwards overlaps the ostium secundum, right? And fuses with it, but there's a orifice in between. This one also not a complete septum, right? So your oval foramen will be formed by the contribution of these two, and that is how the remaining part of the septum primum will become the valve of the oval foramen, right? So when you are writing the answer to this question, supposing it is asked, please remember to mention these names correctly in the correct position, because your ostium primum appears here before the septum primum fuses with the endocardial cushions, and your ostium secundum appears afterwards in the upper part, the secondarily formed foramen, and both these are formed in relation to the septum primum, right? So because when you are writing these things, you mixed up. When you are under stress and when you are hurrying to write these things down, you mix the things. And even though it is very obvious that you have mixed the thing in the hurry, we cannot give you marks for something that is wrong. No, it is written there black and white, wrong. So sometimes when we read your answers, we feel that you knew it, but you have messed it up. But we cannot offer marks for that. So we are always, you have to be careful. And in such instances, if you are drawn this diagram, you will never mix things up. So that is the importance of drawing diagrams in embryology. Right? Okay, so this is the most important part. One other thing I wanted to, here you can see the contributions from different sources. Refresh these things, right? One other thing I want to show you under the cardiovascular system is, of course, we can ask you about aortic arches and development of the vascular system, even though we don't commonly ask. And in relation to these developments, you should be able to explain the congenital anomalies, atrial septal defect, ventricular septal defect, transposition of great vessels, all those different things, right? So if you know the normal development, you can very easily describe those, right? So in any case, if you ask, let's say somebody asks you to write short notes on tricuspid atresia. First, you have to discuss a little bit about the normal development. There again, you don't need to include a lot of details about the formation of heart tube. Just a basic outline of the heart tube and focus more on this area where the defect is. Right? So that is how you are going to answer. Any anomaly that is being asked, you have to write about the normal development a little bit, but focus more on the congenital anomaly. Right? Vice versa, if you ask about the normal development, let's say normal development of atria, then you have to write about the two septa, its normal development, and examples of abnormal developments if it does not take place the normal way. So even if you ask about the normal development or abnormal development, you will have to write both, but the proportions that you are going to write is different. Right? Okay, are you all with me?
Yes, madam.
Right. So I see 63 people are there, 62, but I see 89 names. Right? So there's no purpose of just entering your name to mark yourself present. Right? So you will have to take the benefit of these things whenever and wherever they are available. Just marking your name, marking yourself present will not give anything to you. It will give something for our documentation, but it will not give anything to you. Right? So I hope you're clear about that. I'm telling you these things. People realize all these things after getting repeated. Right? And people come to us with all sorts of questions after getting repeated. And before that, you don't know anything and you don't come to lectures or join the lectures. You just show your presence there, but no active participation, right? So that is my worry, right? Okay, I just wanted to show you the changes at birth. I think it is in the same section, no? So vascular system, just go through the vascular systems and what happens to each aortic arch, right? Circularity changes at birth, yes. Right. So you must have an idea about the fetal circulation, oxygenation that takes place in the placenta, and oxygenated blood is coming via the umbilical vein, right, and drains into the inferior vena cava, right, which contains relatively oxygenated blood now, right. It bypasses the liver to avoid mixing the oxygenated blood in the liver, right, and through the inferior vena cava, it enters into the right atrium, right? So what happens there? It will be directed onto the left atrium via the oval foramen, minimizing the mixing of deoxygenated blood coming from head and neck region. These are very interesting, important phenomena that take place within the human body, right? So it says that these blood streams are separated. They never mix within the left atrium, even though they are in the common cavity. Right? This process is called streaming of blood. In human body, there are certain areas where you see this phenomenon. This is number one. Right? So your blood will be directly collected into the left atrium and it will go into the left ventricle and through aorta, it will be distributed throughout the body. And here there's another important communication. Your deoxygenated blood that is coming from up will enter into the right ventricle, and through the pulmonary artery, it will enter into the lung and come back. So lung is just a solid organ before birth, right? And here there is another site where this blood that is oxygenated, highly oxygenated, I would say, because it is directly coming from placenta through the inferior vena cava from the placenta. And there's no mixing or minimal mixing with deoxygenated blood. And there's small amount of mixing here with the blood that is coming from right ventricle and then distributed throughout the body and ultimately into the placenta. So remember about these sites 1, 2, 3, 4, 5. Important sites where oxygenated blood mixes with deoxygenated blood even though it is minimal. These five sites are very important. And you can compare what happens after birth, changes at birth with the first breath. Right? After the first breath, most of these changes are physiological and it will take 1 or 2 days for them to become anatomical. Right? So with the first breath, the lung expands with oxygen and the peripheral resistance reduces. Therefore, blood flow through the lung increases and oxygenation takes place. Right? The pressure on this side increases, which pushes the valve of the oval foramen towards the oval foramen and closes it. Physiologically at the first instance and anatomically later. Right? And you can get disappearance of ductus arteriosus gradually and replacement of it with the ligamentum arteriosum. And you have two circulations separate now. Right? So your ductus venosus you had in the liver become ligamentum venosum. All those changes that takes place following the first breath. Worth mentioning and important to remember. Right? Somebody might ask you circulatory changes at birth. In a question where we talk about the cardiovascular system or heart or lung or anything. Right? Right. Okay. So we move on to the respiratory system. So it is nothing much for you to explain in the respiratory system. You should know where the respiratory system begins. Actually at the junction between pharyngeal gut and foregut, where you get the respiratory diverticulum and how it can grow downwards and divide in the dichotomous manner. Right? So ultimately behind the trachea and the two bronchi you have the esophagus. So the congenital abnormalities where trachea and esophagus are communicating, tracheoesophageal fistula, are not that rare. Right? Different types of congenital anomalies. Right? So those are the common things that we ask. And remember the fact that your bronchial tree goes on dividing and dividing even after birth. Right? And how many generations that are being formed after birth? Formation of surfactant is important. At which gestational age the formation of surfactant begins. And therefore, you know, surfactant is important for you all to maintain the surface tension. Without the surface tension, the alveoli will collapse. So where do your surfactant come from? Yes? What are the cells that form the surfactant?
The type 2 alveolar cells.
Yes. Particularly these days, you should know this. Neither. Type 2 pneumocytes, alveolar cells කියන්නේ වල pneumocytes කියන්නේ.
Yes.
Alveolar cells තමයි. අනිත් කට්ටිය නිදිද එක්කෙනයිද අහරන් ඉන්නේ?
Madam?
එක්කෙනයිද අහරන් ඉන්නේ අනිත් කට්ටිය නිදිද කියලා හෙව්වේ. Why type 2 pneumocytes are important these days? මම මේ දවස්වල අපි කතා කරනවා type 2 pneumocytes ගැන නැද්ද?
Because of COVID-19.
So what happens in COVID?
They attack type 2 pneumocytes.
Yeah. Okay, to make it look for you to remember, important දේවල් තමයි the periods that where the surfactants are formed. සාමාන්‍ය 7 months වලට කලින් if the baby is born, difficult to survive because of the surfactants. It's not there. Yeah? So type 2 pneumocytes become fully functional around 7 months. If I remember correctly, right? And importance of formation of the further divisions of the bronchial tree after birth. And if you were asked to write about development of the lungs, don't forget to include a few sentences about the development of the pleural cavity as well. Right? It just not just exists there. Right? So we have to little bit talk about the division of thoracic cavity into pleural and pericardial. Right? Okay. We'll move on to the digestive system. Early development of the digestive system, incorporation of the yolk sac, right? So suppose if somebody asks you to write about development of the gastrointestinal tract, then there will be a problem generating inside you because you know quite a lot about development of the gastrointestinal tract, what to write and what not to write. Mandana, we have asked such a question in the past, but anybody anywhere can ask about. So what are the things you are going to write? You can start from the beginning, where the yolk sac is absorbed into the primitive gastrointestinal tract. And do not forget the germ layer where by which it develops, right? And the fact that it is attached to the posterior body wall by the dorsal mesentery and anterior body wall by the ventral mesentery, right? Then the three divisions, okay? Foregut, midgut, and hindgut, and where to where they extend, right? And don't forget to write something about the pharyngeal gut as well, even though we talk about it generally under the head and neck, because it is around the pharyngeal gut that you develop your pharyngeal arches and pouches. And the three vessels that give rise to the separate blood supply: artery of the foregut, artery of the midgut, and artery of the hindgut, right? And derivatives of each foregut, midgut, and hindgut. So basically, if you ask about a general question on development of the gastrointestinal tract, that's all what you have to do, right? Plus the few congenital anomalies, right? And in addition to that, one important thing you have to mention is the physiological herniation of the midgut, because it is an important phenomenon when you compare the rest of the development of foregut and hindgut, right? Physiological herniation and return, right? And when they return, what is the order they come into the abdominal cavity? Because the order in which it returned depends on where they lie afterwards. And there can be congenital abnormalities if they not return in the normal way, right? So that is basically, if somebody asks you about the basic simple question, embryological development of the gastrointestinal tract, those are the things that you are going to write, right? The others, when you separately ask you about each organ, then you know what to write, right? So when you are writing about the stomach, never forget about the dorsal mesogastrium. When you are writing about the duodenum, never forget about the dorsal mesoduodenum, development of the pancreas, right? Right. When you are talking about the stomach, do not forget about the left and right vagal trunks and how they become anterior and posterior vagal trunks in the stomach due to rotation. Right? Is there somewhere in the text? I couldn't locate the exact question. When you're talking about each and every organ, there are certain points that highlight. So try to highlight the important points, right? And formation of fermental bursa or lesser sac, again, a famous question. We have asked this recently, right? How the lesser sac fermental bursa is formed. And its location in relation to the stomach and the stomach bed. And the formation of pancreas. Head of the pancreas occurs at certain level, and tail and body and rest of the pancreas occur a little bit above. That is why when you orientate the pancreas, it has something like this, no? Orientation is something like this, no? So that is why the reason for, positional changes. Sorry. Right. Then formation of the spleen and the two ligaments, gastrosplenic and renal ligament. Right. These two ligaments are very important even for your postgraduate studies. People ask a lot about these two ligaments and how they develop. You know basically how it develops by the development of the spleen in the dorsal mesogastrium. Right. So I'm just highlighting important areas. Harida. And whenever you ask about write about the mental versa or the lesser set, don't forget to draw this diagram. It's very important and it shows the reader or your examiner all the important relations, and you can write your answer by looking at the diagram. These are very easy ways that help you to save time. Okay? Right. So we talked about the anomalies. Nothing much to write about the liver. Liver and gallbladder formation, right? Where the liver outgrowth take place and what are the structures given off by them, right? Your gallbladder, common hepatic ducts, bile duct, all those things, right? And the formation of pancreas, the two buds, right? Dorsal pancreatic bud and ventral pancreatic bud. Where do they come, right? Pancreas also a four-bud derivative. And the ventral pancreatic bud come in close relationship to the dorsal pancreatic bud because of the rotation of the duodenum. And you should know what are the parts formed by the dorsal pancreatic duct and the parts of the duct system that is formed by the dorsal pancreatic duct and the part of the duct system that is formed by the ventral pancreatic duct. Bud. Right? Main duct and accessory duct. And common congenital anomaly. If this rotation takes place in the opposite direction, right? This rotation takes place just generally behind the duodenum. And if this rotation takes place, if the ventral pancreatic bud come to lie close to the dorsal pancreatic duct, migrating in the opposite way, the result is annular pancreas. Not that uncommon. You get duodenal obstruction and the neonate will repeatedly vomit, and you can surgically correct it with much ease, right? Fairly common abnormality, right? Then we talk a little bit about the midgut, physiological herniation, return, and how each and every part come and lie in the abdominal cavity in their respective places. What is the first part that returns back? What is the last part that returns back? Those things are important, particularly for MCQs. Those will be questions, right? So duplication, situs inversus, right? All those things are important. Specifically when you are writing about, let's say, when you are writing about the appendix, the positions, different positions of the appendix, again, worth mentioning. Will you excuse me 1 minute? My battery is low. Wait till I reconnect, right? Right, okay. Sorry for the delay. So we were talking about the appendix. Let's see what are the important areas. Formation of omphalocele, herniation, failure of return into the abdominal cavity following herniation, right? All those things you will be able to explain, right? Meckel's diverticulum, how it is formed, common congenital abnormalities, right? Meckel's diverticulum can sometimes be asymptomatic without giving rise to any symptoms, and it could be an accidental finding during surgery, right? Right. Examples of abnormal rotation of the intestinal lobes and abnormal return, right? Where you can get different forms of abnormal positions. So sometimes your duodenum may be coming anteriorly. So these congenital abnormalities, the importance is, can you hear me?
Yes, madam.
The important thing is the patients can present to you with intestinal obstruction because the gut loops are not normally oriented, right? Right. Yeah. A few, I think last time we have asked this, right? We have asked about the development of the renal canal. No? So some people have drawn this diagram, and people who have drawn this diagrams were able to explain it better. And again, use the correct terminology, right? This is urorectal septum, right? People have given different names to it, right? So initially we have a common cavity, cloaca, where the terminal part of the hindgut and the urogenital system, urogenital sinus, where you have the bladder and the rest of the things, right? And these separate into two parts by the formation of urorectal septum, which comes up and fuses with the cloacal membrane, and the point of division is the perineal body, right? And as a result of this fusion, the two pathways are separated. The urinary bladder on this side, some part of the urethra, and your anorectal canal posteriorly, right? So it's very little that you have to write, but sometimes people confuse with the names and write quite the opposite, right? So that is why I always ask you to draw a simple line diagram and use it to answer your question, right? Which is very much easier because when you draw the diagram, you get the feeling that your book is with you, the reference you have. These things are very easily explained with the diagrams, not with the text, right? Right. Okay, so we'll move on to the urogenital system. There, the importance of intermediate mesoderms comes to place because your genital duct system and the kidney systems develop from the intermediate mesoderm. You should know a little bit about these primitive duct systems, pronephros, mesonephros, that will form part of your female and male duct systems, the mesonephric duct and the paramesonephric duct, and mostly the mesonephric duct, and your paramesonephric duct will develop in relation to the mesonephric duct, right? And their orientation, right? Where your pronephric system is, where your mesonephric system is, and finally your definitive kidney, which is developed from metanephric tissue, right? And how the mesonephric duct develops, right? Collecting duct system of all these unsegmented intermediate mesoderm join and form this duct, which is the male genital duct. It opens into the region of the bladder, and As outpocketing from this duct, you develop the ureter, which penetrates into the metanephric mesoderm that will ultimately become your definitive kidney. Right? So this picture give a clear view about this, and you know that later these will be absorbed into the bladder wall, right? And up to the entrance of the ureters, it will be absorbed into the bladder wall and it will form a triangular area at the back of the bladder wall, which is known as trigone of the bladder. So if somebody asks you about the development of the bladder, part of it is endodermal. The original part of it is endodermal, and part formed by absorbing mesonephric duct, or the trigone of the bladder is mesodermal. Right? So those are the important things that you have to highlight. Right? And the part of the kidney and its duct system that develops from the ureteric bud that is given rise by the paramesonephric duct, and the part that develops from the metanephric tissue itself, right? So up to collecting tubules, they develop from the ureteric bud, right? The rest of the things develop from your metanephric tissue, the renal tubular part. Your proximal convoluted tubule, distal convoluted tubule, all these things develop from here. So I hope you remember this ureteric bud and its duct system has an inductive influence on metanephric tissue, and if only this grows into the metanephric tissue, that these renal tubules will develop, and ultimately both of them unite and perforate to form this final structure. So you should know parts in this system that develop from the ureteric bud and parts of this system that develop from these metanephric renal tubules. Right? Right. And the anomalies: double ureters, ectopic ureters, right? Where the ureter, when the ureter is double, the ectopic one can come up and open at different places, maybe in the vagina, maybe in the urethra, below the bladder neck, again, right? At different positions. So if such things happens, the person will get incontinence of urine, and because urine will leak from there, from the ectopic orifices, right? Okay. A bit about the development of the kidneys. Kidneys develop in the pelvic region and gradually ascends upwards, and the curvature of the body lengthens and disappears, and they gradually ascend upwards, and according to the pathway of travel, aorta will supply it by different arteries, right? Aberrant renal arteries, and sometimes all of them will not disappear and they persist. When they persist only, you call it aberrant renal arteries, right? So if the lower pole of two kidneys are united, it cannot ascend the normal way, right? Because you know your inferior mesenteric artery is there. So the ascent will be arrested, and this anomaly is called horseshoe kidney. Most of the time it will not give rise to any symptoms, but you will come across it as a accidental finding, and you may have seen some horseshoe kidneys in the cadavers downstairs. I hope it's not That uncommon, right? Right. So, bladder and urethra, which parts give rise to the bladder and which parts give rise to the urethra, specifically in males. You have the, prostatic urethra, membranous urethra, and the phallic and penile parts of the urethra. No? So you should know which part originates from which embryological structure. Right. Anything else? And again, when you're talking about male urethra, you should know about this is not embryology, right? Epithelial lining of these different parts, right? And what part of the urethra is narrowest, what part is widest, what is most dilatable, and what is least dilatable. So those are common questions that anybody could ask you, right? So this is the trigone of the bladder, formation by absorbed part of the ureter. And you can see what will happen to the mesonephric duct after this absorption take place. The orientation differs, right? Because these two now come to open into the bladder to different parts separately. Again, at the beginning, it opens through the ureteric bud opens to the mesonephric duct orifice. Now, due to the absorption, you have two separate orifices for the two. Right? Right, genital system, again you know that intermediate mesoderm in the posterior abdominal wall will form the gonadal ridge. Your urogenital ridge is all together. This is the urinary part, this is the genital part. And depending on the genetic constitution of the person, either this, if it is XX, this will develop into ovary. If it is XY, this will develop into testes. And further development of the testes, I hope you remember the formation of gubernaculum and how it goes down into the scrotum, whereas in females, it remains inside the pelvic cavity without going into the perineum, right? Those are again differences between the two systems in males and females, right? So I'm not going to talk in detail about these things. Somebody might ask you the development of the testes, development of the ovary, or compare and contrast the differences, right? Right, the two duct systems, the mesonephric duct and the paramesonephric duct that develops lateral to it, and how it crosses the mesonephric duct and come to the midline and combine with the opposite one, and that is the place where you form the females form the uterus, right? So at the end of development, when the baby is mature, some part of the mesonephric duct remains in females as well. So you should know mesonephric duct is a major duct system in the male, but some part of it remain in the female, giving rise to different small structures. And you should know what the remaining parts are. Similarly, in the paramesonephric duct also, there are remnants that remains in the male, right? So read about those things and you will get familiar, right? More commonly, you can see the mesonephric duct remnants in the broad ligament, right? And abnormal fluid collection on these remnants will give rise to abnormal enlargements, right? So read about these things, right? Right. Molecular changes you need not to know. Yeah. bit about the female duct system, how the uterus is formed, how the vagina is formed, right? There are sino-vaginal bulbs will give rise to the vagina, right? And part of the vagina is actually developed from the paramesonephric duct and rest is from the sino-vaginal bulbs. Right. Development of genital swellings, your urethral swelling and the genital swelling that will give rise to external genitalia. This is the indifferent stage, that there's no difference between the two. And difference takes place in the males and females because in the males it fuses in the midline. That will give rise to the scrotum. And in females it remains open. And all the other morphological differences are due to this fusion and non-fusion. Right? Okay, I think it's about the urogenital system. We'll move on to the last section. I hope it is the head and neck, no? Right. When you're talking about the head and neck, common question is pharyngeal arches and pouches. Derivatives of the second pharyngeal arch, derivatives of the first pharyngeal arch, muscles formed by the second pharyngeal arch, muscles formed by the first pharyngeal arch, the most common questions that anybody could get and anybody could score. Right? So one thing I must tell you at this stage, if somebody asks you the derivatives of the second pharyngeal arch, muscles of facial expression is only one category. Right? So therefore, don't go on writing the list of muscles of facial expression. You have to add the other categories as well. Posterior periopterygastric, stylohyoid, stapedius, auricular muscles. And muscles of facial expression is one category. And if you want, you can give an example or two. Right? So if you want to get all categories, you have to, full marks, you will have to write all categories. Right? Right. So that is pharyngeal arches and pouches is an easiest question if you get one. Very easy to answer and very easy to explain. Right? Yeah. You can see all derivatives here. And you might be asked about the nerve that supply each of these arches and skeletal components that form by each of these. Right? And derivatives of pouches. These are very easy sections. That is why I'm rushing through. Right? Straightforward from the book you can explain. Right. Remember about the inferior and superior parathyroid glands, where they develop, which one has more constant position, which one has more inconstant position, and the reason why. Those might be asked. Right. Lateral cervical cyst, how it develops and why it develops. Right? Common, fairly not that common, but you might come across this. Because of that, somebody might question you. What is lateral cervical cyst and how it develops? Right? So I'm going through, I hope you remember these things. First start syndromes. Right? Pierre-Robin's, Treacher-Collins are right syndromes, right? Where the first starch, derivatives of the first starch are abnormal. Right? And together with the abnormalities in the first starch, there is another congenital abnormality that you will come across within the body. Frost syndrome derivatives are abnormal from the outside of the body when you see it, and there is an associated anomaly, if anybody could remember it, that you cannot see from outside. Anybody who remember?
Tetralogy of
Pardon?
Tetralogy of color?
Yeah, cardiac abnormality, not necessarily tetralogy. Can be any cardiac abnormality. Why? What's the reason? Why facial abnormalities are associated with cardiac abnormalities? In simple terms, if I may ask you. Anybody?
Neural
Yeah.
crystals?
Neural crystals. What happens to neural crystals?
Migration abnormality?
Pardon?
Neural crystal migration abnormality?
Neural crystal migration abnormalities. Why? What is the abnormality? What is the reason? I am asking for the particular association of facial abnormalities with cardiac abnormalities. Your answer is correct, but it's not self-explanatory. From where you get the mesoderm of the face?
Madam, is it because of the
Pardon?
Madam, is it because of the some related to endocardial cushions?
Yes.
So
Were there?
neural crystals.
Somewhere there, but again answer is not self-explanatory. It is because of neural crystals, because endocardial cushions and the septum, more than the endocardial cushions, the septum formation between aorta and the pulmonary trunk. The spiral septum you get in truncus arteriosus. Right? The mesodermal cells there, and the mesoderm of the face, part of the mesoderm of the face come from the neural crystals. And the septum between aorta and the pulmonary trunk, the cells originate from the neural crystals, same origin. That is why facial abnormalities are mostly associated with cardiac abnormalities, septal abnormalities of the heart. Harida?
Okay, madam.
Yes, madam.
Your color kali. Right. Development of the tongue is again a common section. The structures that form the anterior two-thirds of the tongue and structures that form the posterior one-third of the tongue, and how, depending on the contribution of each pharyngeal arch, the nerves of each pharyngeal arch supply the tongue in different manner, right? Anterior two-third, general sensation, mandibular division of trigemina, right? Why is that so? You will have to explain, right? And the special sensation and general sensation of the posterior one-third, why the glossopharyngeal? Why is this happens so? And the most posterior extremity, superior laryngeal branch of vagus, right? You may have to explain the anatomical basis of nerve supply of the tongue. Just like the anatomical basis of nerve supply of the diaphragm, this is again a common question, right? Right, thyroid gland again, it's a very common question, and most of you answer well for if we ask about the development of the thyroid gland. Don't forget to write about ectopic thyroid, thyroglossal cyst, right, which is a midline cyst, so on and so forth, even if you ask about the normal thyroid gland, right? So when you're talking about the thyroid gland, this migratory pathway is important. and during which time different cell types appear here is important, and during which time they start secretion is also important, right? And the fact that even after removal of the thyroid gland, if the symptoms of some thyroid related disease persist, you will also have a look at ectopic thyroid gland, and you have to look along its migratory pathway. And that is why we learn all these things. Right? Okay, that is about the thyroid. Development of the phase, the 5 processes, right? And the structures that form by each of these processes, again, a common area, right? For example, if you get these medial nasal prominences, part of the nose and part of the lip formed by them. Part of the nose that is formed by lateral nasal prominences. Right? You should know all these. Yeah. You can see all these things in a nice way. Now, other than the diagrams of this book, the other thing you have to refer is the tables. It will nicely explain you summarized data on development, not only in the region of head and neck, all the other sections, right? Intermascular segment, a little bit, and parts of parts that is formed by the intermaxillary segment, philtrum of the lip, and the part of the jaw that contain the upper 4 incisor teeth and the primary palate, right? And how your secondary palate develops, and the common abnormality, cleft palate, anterior defects, posterior defects, combination of defects, revise all these things, right? And together with that, learn a little bit about the development of the nasal septum, how it comes and fuse with secondary palate to give rise to 2 separate cavities. And in this book, development of the nose is not given under single heading. Part of it will be dealt with the facial prominences, right? The outside of the nose. Inside of the nose is dealt with the palate, right? So when you have to write about, somebody asks you about the development of the nose, you will have to combine all these things and write a single answer. You will have to talk about the external appearance of the nose, formation of the nasal septum, fusion of it with the palate, and also formation of paranasal sinuses, which are equally important, right? And as a congenital abnormality, you can mention cleft palate as well, because there will be open communication between the nose and the oral cavity if there's failure of fusion of palatine shells, right, of the maxillary processes, right? Different types of defects that you will get, anterior defects, posterior defects, and the combination of them, right? Again, cleft palates and lips are common, right? So to that question about the nose, this is the final bit you have to add it, right? The presence of oronasal membrane posteriorly that needs to be perforated to form the posterior nasal cornea, right? Or definitive cornea. That's the final part of its development. So you will have to combine all these things when you ask about the development of the nose. Okay?
Yes, madam.
Right. So we don't talk about the development of the teeth, and I don't think that we might ask you. We don't give lectures on development of the teeth as a separate one, no? So it might be as a part of other lectures, but singularly we will not focus on this in general, right? If you know about primary and secondary dentition, that would be enough, right? And your teeth, primary dentition will loosen as a formation of secondary dentition. A few things, few important things, that would be enough. Other than that, we might not ask you it as a separate question, right? Anything else? We have to talk a little bit about the development of the ear. And this is again nothing much. You know where the external auditory meatus will develop and where the internal auditory meatus develop and where your tympanic cavity develops, right? So this is the most important diagram in relation to the development of the ear. External auditory meatus from the first cleft and tympanic cavity, right? And auditory tube and formation of ossicles in the mesenchyme. All these things you can just explain if you have a diagram like this with you, right? And I don't think that we might ask you about details about the development of the inner ear. The things that you know for Ross anatomy would be enough. The membranous labyrinth, bony labyrinths, and their orientation, right? Little bit about the anomalies of the external ear. External ear developed by about 6 or 7 mesodermal proliferations, right? Therefore, external ear is the pinna or the ear lobe is different in different individuals, depending on the contribution of these mesodermal masses, right? And ear abnormalities are closely associated with renal anomalies, right? So therefore, an abnormality in the earlobe will be sometimes, not always, it might be telling you that there's a problem in the kidney inside, right? Your book mentioned somewhere about that association, right? So nothing much about the ear. And about the eye, what you have to mention is it develops as outgrowth of the forebrain. There are 2 optic vesicles or optic grooves at the beginning that grows towards the ectoderm. And you can see an area within this ectoderm that is differentiated into lens, right? And this part will come to lie close contact with the future lens, and finally formation of the optic cup. And I hope you remember I told you once, the outer layer and inner layer of optic cup, right? And the 2 layers will the neural and pigment layers of the retina, and how they fuse later, and why retinal detachment takes place between these 2 layers, right? Right. The other picture is not there, no? I don't know. Might not have downloaded. Yeah. Doesn't matter. Right? So not very much to remember. A little bit about the pupil and the iris and the muscles. Constrictor pupillae and dilator pupillae. Right? I don't think that rest of the things will be asked other than the basics about the development of the eye. One thing I want to highlight here is, sorry about that, I couldn't tell you earlier, the lens and the retina develop from the ectoderm, right? Retina from the neuroectoderm and lens from the surface ectoderm. Any structure in our body that we make contact with the outside world develop from ectoderm, right? Maybe let it be the skin, epidermis, nails, right? Sensory nerve endings, your entire central nervous system, all those things develop from the ectoderm. If you go through the list of structures that develop from ectoderm, endoderm, and mesoderm, you can again refresh this, right? So about the central nervous system, what you have to discuss is formation of your neural tube. Firstly, you get the neural plate, right? Again, some part of it's missing. Then it becomes a neural groove. Don't forget the neural crest cells. And the two ends of the crest, the groove, unite in the middle and form the tube. And tube separate from the surface and come down. And neural crest cells separate and form two segments on either side of the neural groove, right? And ultimately they will migrate into different areas, right? So basic description about formation of neural plate, neural groove and neural tube has to be described wherever you get a question about the embryology of the nervous system. But the extent you are going to describe it differs from place to place, right? And this shows a cross section of your neural tube where you have those different types of cells, matrix cells that later differentiate into neuroblasts, so on and so forth. And don't forget about the formation of velar plates and basal plates. If you were asked about the development of any place within the central nervous system, you have to talk about these two, right? So depending on what structure you're talking about, their fate is different. Because when the ponds develop by opening up of these structures, the orientation of velar plate and the basal plate varies, right? Position. Above downwards orientation will become lateral and medial orientation, right? Right. Revise what you have learned about different types of neurons, morphological differences of neurons, and with the examples where you find each of them, and neuroglial cells, right? Astrocytes, oligodendrocytes, all different types, right? Again, remember the fact that the positional changes of the vertebral column, spinal cord in relation to the vertebral column at birth, where the location is, and what's the location during adulthood. And the myelinations of the central nervous system is not completed at birth. It goes on up to an early stage of your infancy and childhood. Those are important facts that you have to remember, right? The rest is a bit about development of the ventricular system and congenital anomalies closely associated with spina bifida, and meningocele, meningomyelocele, and anencephaly, plus abnormalities in the ventricular system and hydrocephalus. Right? So this is what I have told you. When it opens up, the orientation of velar plates and basal plates differ. Right? So if you know the basic, there is nothing much to remember actually, because if you know the basics and facts, then you can remember all the other things if you know the embryological basis of most of the things. Right? So I think I have gone through the entire extent, highlighting you different areas of importance, right? Before reading your embryology again, just go through the diagrams in any section. Before answering the question, you might be practicing answering questions these days now. So what I advise you is any system anywhere, just go through the diagrams and try to see whether you can explain the story in your own words. Don't try to by heart the words in the book, right? So that way you can survive and it will be easier to remember. Okay? Shall we stop there? Okay, it's
Thank you, madam.
Okay, madam.
right. I hope this has been useful. I couldn't find any other way to revise this. Right? So let's see how useful it has been, right? So my next revision class, I think, towards the end on pelvis and perineum. I'll put up 2 questions. You have to answer the 2 questions and come for the discussion, right?
Okay, madam.
Okay, right, we'll finish.
Thank you, madam.
Thank you, madam. Thank you.